Your body felt older this morning than it did when you went to sleep last night. Not older by years, older by decades. You swung your legs off the bed, stood up, and for a few seconds, maybe longer, you moved like a machine that had been left out in the frost. Stiff, creaky. Joints that needed a moment to remember what they were designed to do. You probably blamed age, or a bad mattress, or just how it is. But here is what was actually happening inside your body while you slept. While you lay still for 7 or 8 hours, your connective tissue, the microscopic scaffolding that wraps around every single muscle, organ, nerve, and cell you own, was quietly doing what it always does during stillness. It was weaving, filling the empty space between your fibers with a fine, sticky mesh of collagen threads. Randoms, disorganized, binding things together that were never meant to be bound. Scientists who study fascia under a microscope have a name for this material. They call it the fuzz. And every morning, the stiffness you feel is your body asking you to do one thing. Move enough to tear that fuzz apart before it sets permanently. Because here is the part that changes everything. That morning stiffness? It is not a minor inconvenience. It is not just tightness. It is a live demonstration, running in real time, of the exact physical process that ages your body at the cellular level. Aging is not primarily a chemical countdown. It is a structural one. It is your body's internal scaffolding, slowly cross-linking, dehydrating, and stiffening, until the fluid that feeds your cells cannot reach them, and the waste your cells produce cannot leave. And the mechanism that reverses it is not a drug. It is not a supplement. It is not a genetic intervention. It is a stretch. Not because stretching loosens muscles. That is the shallow answer, the one that misses the physics entirely. Stretching reverses aging because it is a mechanical force applied to living tissue, and living tissue has no choice but to respond to mechanical force at the molecular level. When you stretch, you do not just lengthen a muscle. You change the physical pressure inside the tissue. You squeeze stagnant fluid out. You create a vacuum that pulls fresh, nutrient-rich fluid in. You physically deform cell membranes, which opens pressure-sensitive channels, which triggers the cells inside to break down old rigid collagen and lay down new elastic fibers. This is not a metaphor. This is the physics of tissue remodeling, a field called mechanotransduction, and it explains something that should bother every person who takes vitamins, watches their diet, or does anything at all in the name of longevity. You can take every anti-aging supplement that exists. You can eat the most perfectly optimized diet on the planet. But if your extracellular matrix, the fluid-filled web your cells actually live inside, has cross-linked into something closer to leather than living tissue, those nutrients physically cannot reach your cells. The delivery system is blocked, not slowed. Blocked. The question is not whether you are aging, you are. The question is whether the cage your cells are sitting in is made of flexible, living tissue or hardening cement. There is a surgeon named Gil Hedley who has spent decades studying the human body not through scans or blood work, but by hand, dissecting cadavers layer by layer and filming what he finds between the structures most anatomy textbooks bother to label. In one of his must-watch demonstrations, he peels back the skin from a body that lived a sedentary life, and between the skin and the muscle beneath it, he finds something that looks almost biological in the wrong direction. A thick, whitish, cobweb-like film, matted, dense, clinging to everything. He calls it the fuzz. And he explains calmly and without drama that in a body that moved regularly throughout its life, this layer is thin and wispy, barely there. In a body that did not, it accumulates. It thickens. It weaves itself into the gaps between muscle layers, between organs, between the fibers that were never meant to be connected. It bonds structures together that need to glide freely across each other to function. This is not a disease. It is not a pathology. It is just what collagen does when there is no mechanical force telling it to organize itself differently. Every night, while you are still, your body lays down new collagen in the spaces between your tissues. Without movement, that collagen has no mechanical signal guiding it into organized parallel fibers, so it lands randomly, like picking up a handful of dry spaghetti and dropping it on a wet surface. It sticks wherever it falls. And when morning comes and you stand up and feel that familiar resistance in your hips and lower back and neck, you are feeling the accumulated overnight deposit of disorganized collagen, the fuzz being torn apart by your first movements of the day. The problem is that for most people, those first movements are not enough, and over years and decades, what the morning routine fails to clear continues to build. To understand why that matters beyond mere stiffness, you need to understand where your cells actually live. Most people carry a mental image of the body as something like a machine, solid parts connected by cables, running on chemical fuel. But that image misses what may be the most important structural reality in human biology. Your cells do not live in solid tissue, they live in a fluid. A rich, dynamic, protein-dense fluid called the extracellular matrix, the ECM, that fills every space between every cell and every tissue in your body. Think of it as a biological ocean, not a passive one. An active, living scaffolding made of water, proteins, and collagen fibers, constantly exchanging nutrients flowing in and waste flowing out, constantly receiving signals, constantly being remodeled in response to pressure and movement. Every cell you own, muscle cells, nerve cells, immune cells, the cells lining your blood vessels, is embedded in this ocean. And the health of the cell is almost entirely dependent on the health of the ocean it sits in. When the ECM is fluid, dynamic, and well hydrated, nutrients move freely to the cells. Waste moves freely away from them. Cellular signals, the biochemical messages that tell your immune system to activate, your tissues to repair, your cells to divide correctly, travel through this medium the way sound travels through water. Efficiently, completely. When the ECM begins to stiffen and dehydrate, all of that slows. Then it stops. And here is the mechanism that causes the stiffening. It is not mysterious. It is chemistry, specifically a process called molecular cross-linking. Collagen, the structural protein that forms the fibrous backbone of the ECM, is designed to exist in long, parallel, organized strands. Like a rope, individual threads laid alongside each other in the same direction, creating something that is simultaneously strong and flexible. The organization is what gives it those properties. A well-organized rope can bear enormous tension without breaking and return to its original shape when the tension releases. But collagen does not maintain that organization on its own. It requires mechanical input. It requires the physical signals that come from movement, from muscles pulling on tendons, from joints loading and unloading, from tissues being stretched and compressed and released throughout the day. Those mechanical forces are what tells collagen fibers to lay down parallel to each other, to stay separated, to remain organized. Without that input, the chemistry changes. Collagen molecules begin forming bonds not just along their length but across to neighboring fibers. Perpendicular bonds, random bonds. The kind of bonds that form not because the tissue is being built toward anything, but because cross-linking is simply what unstimulated collagen does in a warm, wet, biochemically active environment. Imagine spilling a bottle of wood glue onto a pile of loose threads and letting it dry overnight. No one arranged the threads. The glue did not follow any plan. It just hardened around whatever it touched. The threads are now bonded to each other in a completely random pattern, and trying to pull them apart without tearing them requires force the tissue was not designed to generate. This is happening in your body right now. It has been happening every night for years. And the sedentary hours, the desk, the couch, the car, the commute, are adding crosslinks during the day too, not just while you sleep. The body reads a lack of mechanical input not as rest, but as a signal to reinforce, to consolidate, to make the tissue denser, less pliable, more resistant. The body's logic is ancient and straightforward. If this structure is not being loaded, it must not need to move. Make it stable, stiffen it. Save the metabolic cost of maintaining flexibility that nothing is demanding. The consequence is tissue that begins slowly and incrementally to behave less like living muscle and more like old leather. There is a useful way to feel this principle without a microscope. Pick up a dry sponge, one that has been left out, it is completely desiccated and rigid, and try to bend it. It does not flex, it resists, and if you apply enough force, it cracks. The structure is intact, the material is the same, but without fluid, the mechanical properties have transformed entirely. Now run that same sponge underwater, squeeze it several times, let it absorb the fluid, and suddenly it is pliable, resilient, able to compress and spring back without damage. Your fascia and your extracellular matrix behave the same way. Hydrated tissue is elastic. Dehydrated, cross-linked, mechanically unstimulated tissue is brittle. And the reason unstretched tissue dehydrates is not simply that you failed to drink enough water. It is structural. Compressed tissue physically repels fluid. The internal pressure created by cross-linked, tangled fibers squeezes the ECM into a state where new fluid cannot easily enter. The water that was there gradually leaves. The tissue becomes denser, less permeable, more compressed. and denser tissue compresses the capillaries running through it, the microscopic blood vessels delivering oxygen and nutrition to the cells embedded in the ECM. This is the cascade that most people never connect. Stiffness is not the end of the problem. Stiffness is the beginning of a circulation problem, a nutrition problem, a cellular waste problem, a cellular aging problem. The cells sitting inside that compressed, dehydrated, cross-linked tissue are not receiving what they need, and they are not expelling what they produce. They are sitting in an increasingly toxic, nutrient-poor environment inside a structural cage that is slowly tightening around them. And this is where the physics becomes extraordinary, because the solution is not pharmaceutical. It is mechanical, and the mechanism is so elegant that it reframes what stretching actually is, at a level most people have never considered. When you apply a sustained stretch to a tissue, you are not just lengthening a muscle. You are applying a physical force to a three-dimensional biological structure that is filled with cells. Those cells have membranes, and embedded in those membranes are proteins called mechanosensitive ion channels, physical structures that respond not to chemistry but to deformation, to pressure, to being pulled. When a stretch deforms the tissue, it physically distorts the shape of the cell membranes inside it. That distortion opens the mechanosensitive channels. Ions flow across the membrane. The electrical potential of the cell changes. And that change in electrical state triggers a cascade of genetic activity inside the cell. The cell reads the mechanical force as a biological instruction and begins transcribing genes that it would otherwise leave dormant. Specifically, it begins producing matrix metalloproteinases, enzymes whose job is to cut through and dissolve old, disorganized, cross-linked collagen. The molecular glue that accumulated during weeks and months of stillness begins to be enzymatically cleared. And in the cleared space, guided now by the mechanical tension of the stretch, new collagen fibers are laid down in organized, parallel, functional alignment. The cell took a physical force and translated it into chemical biology. It took a stretch and turned it into a tissue remodeling command. This process has a name, mechanotransduction. And it is not a theory or a hypothesis. It is one of the most well-documented phenomena in cell biology, studied across dozens of tissue types in hundreds of peer-reviewed studies. Cells are not passive recipients of chemical signals. They are mechanical sensors. They read force, they read pressure, they read deformation, and they respond to it by changing what genes they express. This means that when you stretch, really stretch, with sustained, deliberate load held long enough for the tissue to respond, you are not performing a flexibility exercise. You are issuing a command at the genetic level. You are telling your cells to dissolve their own cage and rebuild it in a form that serves movement instead of resisting it. You are using physics to change chemistry. You are using mechanics to alter biology. The body you have at 60 is not simply the product of time passing. It is the product of the mechanical inputs your tissue received, or did not receive, across those decades. Time did not make your tissue stiff. Stillness did. And what stillness built, movement can dismantle. Not all at once, not in a week, but systematically, consistently, and at a cellular level that no supplement can replicate, because no supplement can open a mechanosensitive ion channel. No pill can deform a cell membrane and trigger matrix metalloproteinase production. No injection reaches into the ECM and physically breaks a cross-linked collagen bond. Think about what happens when you squeeze a wet sponge over a sink. The compression forces the fluid out. Every drop that was held inside the structure is expelled by the mechanical pressure of your grip. Then you release it. And the sponge, now decompressed, draws new fluid back in, not because you poured water on it, but because the release of pressure created a suction effect. A vacuum. The fluid moves in to fill the space that the decompression opened. Your tissues work on exactly this principle, and it is more important than almost anything else in this entire conversation. When you hold a sustained stretch, you are compressing one side of a tissue and elongating the other. The mechanical pressure of that stretch squeezes the interstitial fluid, the fluid living inside the ECM, out of the tissue and back toward the lymphatic and circulatory systems. That fluid is carrying something with it when it leaves. It is carrying metabolic waste. The byproducts of cellular metabolism, carbon dioxide, lactic acid, inflammatory cytokines, cellular debris, that have been accumulating in stagnant, unstretched tissue, sometimes for days or weeks, are physically expelled by the mechanical compression of a stretch. This is not a slow, passive, diffusion-based process. It is a pump. A mechanical pump, operated by movement, that clears the cellular environment of waste that biochemistry alone cannot efficiently remove from compressed tissue. And then you release the stretch. The decompression phase, the moment you let the tissue return toward its resting length, creates a pressure differential. The tissue, now temporarily lower in fluid pressure than the surrounding circulatory system, draws fresh fluid back in. Fluid that is rich in oxygen, glucose, growth factors, and the raw materials cells need to repair and replicate. The cells that were sitting in a stagnant, waste-saturated environment moments ago are now bathed in fresh supply. The exchange that the circulatory system was struggling to accomplish through compressed capillaries is suddenly completed mechanically, in seconds, by the simple physics of squeeze and release. This is why people who stretch regularly report feeling genuinely different, not just more flexible, but more awake, less foggy, physically lighter. They are not imagining it. The fluid dynamics of their tissue have changed. Their cellular environment has been refreshed. Their cells are, quite literally, better fed and cleaner than they were before the stretch. But the fluid pump effect is only part of what is happening, because the collagen fibers being stretched are not electrically neutral. And what they do, when placed under mechanical tension, takes the biology into territory that most people have never been told about. Collagen is a piezoelectric material. Piezoelectricity is a property of certain crystalline structures, the ability to generate an electrical charge in response to mechanical stress. Press on a piezoelectric crystal, bend it, stretch it, and it produces a measurable electrical current. No battery required, no external power source. The mechanical deformation itself generates the electricity. Quartz does this. Certain ceramics do this. Bone does this, which is part of how bone remodels itself in response to load, a process that has been studied extensively in orthopedics. And collagen does this. When you stretch a collagen fiber under sustained mechanical tension, the physical deformation of its crystalline structure generates a small but real electrical charge along the length of the fiber. That charge propagates through the surrounding ECM, the fluid medium that, as you now know, every cell is embedded in. The cells pick up that electrical signal through their membranes, and they respond to it the way cells respond to all electrical signals, by changing their behavior. Specifically, the piezoelectric current generated by stretched collagen acts as a directional instruction to fibroblasts, the cells responsible for producing new collagen. It tells them not just to produce collagen, but to produce it in alignment with the direction of the mechanical tension. Organized, parallel, functional. The exact opposite of the random cross-linking that accumulates during stillness. This means that stretching does not just break down old, disorganized tissue. It simultaneously signals the construction of new organized tissue, and it uses electricity generated by the stretch itself to deliver that instruction. The body is using physics to guide its own reconstruction. The mechanical force of the stretch generates an electrical current, and that current tells the cells exactly where and how to build. There is no pharmaceutical that replicates this. There is no supplement that generates a piezoelectric charge along a collagen fiber. There is no intervention in the entire landscape of anti-aging medicine that reaches into the structural architecture of the ECM and issues electrically guided construction commands to fibroblasts. Movement does this. Specifically, sustained mechanical tension does this. And it has been doing it since the first multicellular organisms evolved connective tissue, hundreds of millions of years ago. Now consider what happens when this signal is absent for years. The cells inside chronically stiff, cross-linked, compressed tissue are not simply undernourished, they are under physical stress. Continuous, unrelenting physical stress, the kind that comes from being mechanically compressed by the tissue surrounding them, day after day, without the periodic decompression that movement provides. And cells under sustained mechanical stress respond in a specific, well-documented way. They age faster. There is a cellular state called senescence, sometimes called the zombie cell state, where a cell stops dividing, stops performing its normal function, but does not die. It lingers. And it does something damaging in the process. It secretes inflammatory signals into the surrounding tissue, a phenomenon researchers call the senescence-associated secretory phenotype. Senescent cells are essentially alarm sirens that never turn off, continuously broadcasting a low-grade inflammatory signal that degrades the tissue around them and accelerates the senescence of neighboring cells. What triggers cellular senescence? Several things: telomere shortening, DNA damage, oxidative stress. These are the mechanisms most people have heard about in the context of aging. But there is another trigger that receives far less attention: sustained mechanical compression. Cells that are physically compressed, trapped inside a rigid extracellular matrix with no mechanical relief, accumulate structural damage in their cytoskeleton. Their nuclei deform. The physical architecture that the cell depends on to divide correctly is compromised, and the cell reads that structural damage as a signal to stop dividing and start broadcasting distress. Chronic stiffness is not just uncomfortable. It is creating the physical conditions for accelerated cellular aging, happening silently inside compressed tissue, completely beneath the threshold of conscious awareness. You do not feel a cell become senescent. You feel the downstream consequence, the joint that never quite recovered, the chronic, low-grade inflammation your doctor cannot explain, the fatigue that sleep does not resolve, the slow narrowing of the range in which your body feels comfortable moving. And running underneath all of it is the circulatory consequence that ties everything together. Capillaries, the smallest blood vessels in the body, the ones that perform the actual work of delivering oxygen to tissue and removing carbon dioxide from it, are not rigid tubes. They are elastic, pressure-sensitive structures embedded in the same connective tissue matrix that cross-links and stiffens with disuse. When the surrounding ECM compresses, the capillaries compress with it. Their diameter narrows. The volume of blood they can carry decreases. The contact time between blood and tissue, the window during which oxygen and nutrients can diffuse across the capillary wall and into the ECM, is reduced. Open, hydrated, flexible tissue does the opposite. The capillary beds inside supple, regularly stretched fascia are uncompressed. They carry full blood volume. The surface area available for nutrient exchange is maximized. The cells living in that tissue receive their full allotment of oxygen, glucose, and growth factors, and they expel their full load of waste. The difference in cellular health between well-perfused tissue and poorly perfused tissue is not marginal. It is the difference between a cell operating at its biological potential and a cell operating in a state of chronic, low-grade deprivation. Flexibility at this level of analysis is not a fitness quality. It is as a circulatory quality, a cellular nutrition quality, a longevity quality. The suppleness of your connective tissue directly determines the efficiency of the circulatory system operating inside it. And the circulatory system inside your tissue directly determines whether your cells are aging at the rate your calendar suggests or faster. So what do you actually do with this? The answer is simpler than the science that supports it. And that simplicity is important, because complexity is the enemy of consistency, and consistency is the only thing that works at the structural level. The research on tissue remodeling through mechanical loading points to one clear principle: the stimulus that triggers meaningful change is not intensity, it is duration. Short, aggressive stretches, the kind performed as a 30-second afterthought at the end of a workout, do not hold tissue under tension long enough to trigger the mechanotransduction cascade, activate piezoelectric signaling, or allow the fluid pump to complete a full exchange cycle. The tissue resists briefly, and then the stretch ends. Nothing structural changes. What changes tissue is sustained, low-load tension held for time. 2 minutes per position is the threshold that appears repeatedly in the literature on fascial remodeling. Not painful, not aggressive. A gentle, deliberate pull, enough to feel the tissue engage, not enough to trigger the protective contraction reflex, held for a full 2 minutes while breathing slowly and allowing the nervous system to release its guarding response. 2 minutes for the hip flexors, 2 minutes for the thoracic spine, 2 minutes for the hamstrings and posterior chain, 2 minutes for the pectoral fascia and shoulder capsule. Not as a workout, as a daily physical maintenance practice. The mechanical equivalent of brushing your teeth, performed not for athletic performance but for structural survival. Done consistently, this protocol does 4 things simultaneously. It breaks down the overnight crosslinks before they consolidate. It activates the fluid pump, clearing waste and drawing fresh nutrition into the tissue. It generates piezoelectric signals that guide organized collagen reconstruction. And it mechanically decompresses the cells inside the tissue, reducing the structural stress load that drives senescence. None of this requires equipment. None of it requires a gym membership, a trainer, or an hour of free time. It requires a floor and 2 minutes of attention per position, applied with enough regularity that the structural cross-linking never gets ahead of the structural clearing. The body you are living in right now is not fixed. It is not a declining machine running down a predetermined biological clock. It is a mechanical system that responds at the cellular level, at the electrical level, at the fluid dynamics level to the physical inputs it receives. Give it stillness, and it consolidates around that stillness. Give it sustained, deliberate mechanical tension, and it dissolves what stillness built and reconstructs something more alive in its place. The morning stiffness you woke up with today is not a verdict. It is an invoice, and the currency it accepts is remarkably cheap.